And oh, the meeting has, okay, sorry. And I'm going to share with you what we've been doing so far. So I came in when the pyrolysis was bought. So we started everything from the scratch, trying to develop methods, trying to get staff to work for our group. So I will not do much introduction because we all know about microplastics, but I will just start by introducing ourselves to plastics. So we all know that globally, the production rate of plastics has been increasing since 1950, reaching 348 million tons in 2018. But the simple question I ask myself, and I think all of us do ask ourselves, is what happens to all the plastic that we produce? Well, a simple answer we all know is that they do get into the environment some way, somehow. And then with time, all of these plastic products that get into the environment will break and become smaller and smaller to become what we call microplastics, which are plastic particles that are less than 5 millimeters in size. Now, currently, as we study plastics, we know they are in every metrics that we've analyzed, which includes air samples, which include dust, seafood samples, food, drinking water, beers, wastewater, biosolid, rain, name them. There have been a couple of places that plastic has been actually been identified. Now, currently, there are several techniques that are used to analyze plastics. Now, the most common ones that we all know or that have been published in literature include using density separation methods after digestion of the plastics or the organic content in these samples, and then using the density separation method to actually separate the plastics onto a filter paper. And then some labs go ahead by, let me see, there's so many messages coming. I don't know whether it has to do with me. Is the sound all right for everyone? Okay, sorry.
I think, no, it's just, it goes in and out slightly. We can still hear you, but it fades in and out a bit. So
let me try and connect the headphone. Maybe that will make things better. That's okay. Is this better?
Yeah, it sounds okay. I'll let you know if anything goes. So, thank you.
Okay. Yeah. So some studies actually use optical microscope to actually count the particles on these filters. Some have used infrared to stain these particles. Some actually go ahead and pick some of these particles, either percentage-wise, or they analyze all the plastic particles that they assume to be on these filters using either FTIR or Raman spectroscopy. Now, the good thing is that these techniques or these procedures actually gives us the number of particles. It gives us the sizes of the particles, the color, the shape. However, it does not give us the mass of plastic particles that are in one sample. Now, these techniques are also operator dependent. So I can say one particle is a plastic. Holly can also come tomorrow and say this is not plastic based on their assumptions and how they think the identification should be done. Now, these techniques are also size dependent. Size dependent in the sense that now, depending on the filter size you use, the pore size of the filters that you use, it's either you use 22 micron, 1 micron, whatever micron you use, definitely there will be a big or a large fraction of the particles that will be lost that you are not going to identify. And the techniques that are also used to identify the polymer types, which include the Raman and FTL, are also size dependent. I think Raman can go up to 1 micron, and then FTL can go up to, I think, 20. Based on different literatures, I've seen people are saying different sizes that they measure in their labs. Now, to actually go through all this process, it's time consuming. Of course, you have to digest, you have to count, you have to do a density separation. We've tried all this in the lab, and I can tell you, it's very hard to go through all these techniques. Now, the interesting thing is that, like I already mentioned, there's increasing challenges or uncertainties with the amount or the size of plastics that we identify using all these techniques. It's very hard to go to the nano range. And like I said, FTIR can go above 20, I think above 20 micron. And then macro Raman can also go, I think, above 1 micron. I stand to be corrected because different labs report different things and different labs are saying they can go below some of these sizes. Now, Pyrolysis GCMS has its own advantages and disadvantages. So the good thing is that it's a suitable tool for simultaneous identification and mass quantification of plastics. Now, what I mean by this is that you can actually have one sample and then put a sample in a pyrolysis tube and then get different polymers identified using just one run, compared to FTR, where you have to actually pick the particles one by one and try to identify them individually. With pyrolysis, once you analyze one sample, if you have 10 polymers, once you've run the standards, you can actually identify all these polymers in one sample based on the instrument sensitivity and the limit of detection. Now, with pyrolysis GCMS, you can also overcome preparative manipulations of the sample. It requires small sample size with little or no preparation. It also enables quantitative estimation of the mass of plastic irrespective of the particle size or shape. Now, pyrolysis GCMS also are independent, like I just said, independent of the particle size, color, shape, or the numbers, and allows a cross actually comparison of data. It also reduces the sample treatment stages that you go through. Now, one will ask, how does pyrolysis work? Now, pyrolysis is a destructive technique, of course. With FTR and Raman, you can actually keep the polymers and reanalyze them for other stuff in future. But with pyrolysis, once you analyze the sample, everything that you have in there is gone because it's a destructive technique. What I've seen people do is that, which we also try to do, people try to identify the polymers like using FTIR, Raman, and then they pick a fraction of these particles and then analyze with pyrolysis GCMS. So in that way, the fraction that they lose through pyrolysis GCMS, they don't care because they have some fraction that they can still analyze with FTR and other. Now with pyrolysis, you introduce a sample into a microfenix at a high temperature. So we have single shot pyrolysis and we have double shot. With a single shot pyrolysis, you set a known temperature, which is like, in our lab, we use like 650 degrees. So it's called flash pyrolysis. You drop a sample into the microfenix chamber, and then whatever sample you have or whatever polymers you have are decomposed. And then this actually goes through the GC column, and then they're separated, and they detect, you just see this chromatogram here. So after the sample is injected into the GC, it goes to the detector, and then you have this chromatogram or pyrogram, which then contains all the polymers that you have in your sample. Then the next stage is where you have to actually go in and manually screen to know which polymers you have. So in our lab, we use indicators. Everyone using pyrolysis will use an indicator. So like for polystyrene, for instance, we have the monomer. It breaks into monomer, dimer, and the trimer. Then you try to look for the monomer, dimer, and the trimer in your sample to actually confirm that this is polystyrene. So what we do is that you don't actually use just one break product of the polymer. You try to look for other products to actually confirm the polymer type. So this is a simple extract that we got. This is PVC, this was PP, this is PE, PMMA, and PS. And like I said, this is just one sample. And one sample analysis can give you all these 5 polymers if you've done your method development and you have your standards actually run. So in our lab, after developing the pyrolysis GCMS method, we wanted not a fast, but more a way to go about the sample pre-treatment, try to reduce the amount of work that goes in. And then we thought about pressurized liquid extraction. This is something that is used in most labs to extract organic compounds. So what we did was that we tried to develop a method. There was already a published method on this, where the person actually combined the extract from the pressurized liquid extraction, evaporated the solvent, and then analyzed it using the FTIR. So we thought about combining these two techniques, because if pyrolysis can be used to actually identify polymers or simultaneously identify and quantify polymers in a sample, if you have a technique that can then be used to extract all the polymers in your sample, then you have a solution to actually just inject and then get all these samples analyzed, instead of filtering onto a filter paper and trying to cut into pieces and roll it into the pyrolysis. So we did a method, and then we found out that dichloromethane at 180 degrees and 1,500 PSI can actually extract 7 plastics. So we have polystyrene, polyethylene, PET, PP, PVC, PMM, and PC. Now, I have to say here, polypropylene, polyethylene, and PVC has its own solubility issues. So what we did was that we knew this before we started the extraction. We knew they were going to crash out at room temperature. So what we did was that we did a stability test to check at which times or within which minimum hours can you have an extract from the ACE and then still inject and still have good, still not have the plastics actually crashed out in your solution. So the test, I didn't show the data here, but the test shows that if you extract from the PLE, within 2 hours, you can still analyze and get a good representative data. So what we did was that we extract with PLE, and then we inject 80 microliters onto the pyrolysis. Now, in our lab, in order to reduce sample pretreatment, like doing a digestion, trying to do density separation, we use a double shot pyrolysis, like I was explaining earlier. So we have a single shot where the sample is dropped into high temperature. It breaks every product you have in there, including the additives, organic compounds. Then we have the double shot where you analyze one sample twice. So you have the sample analyzed with a low temperature to actually thermally dissolve the organics, the additives you have in your sample, and then the plastics can be analyzed in the second shot at a high temperature. The idea is that you can use the low temperature to actually just get rid of the organic matrices that you have in your sample, and then you analyze the plastics in the second shot. So this technique actually enables mass concentration using external calibration. So this is what I'm talking about. So this is an example of a single shot. I don't know, Holly, is the Keza working for people to see? Okay, good. So this is a single shot of a biosolid sample at 650 degrees. So this was when we analyzed an extract with a single shot flash pyrolysis. It goes in there for 650 degrees. You have the additives, all the organics, and all the polymers being extracted here. Now with a double shot, this is the first shot of the double shot. So what it means is that we analyze the same sample with a low temperature first to get rid of the interferences, and then the polymers are then analyzed in the second shot. So with the same sample, if you use the first shot to clean, then you get a cleaner pyrogram to then quantify your polymers in there. Now this is a simple total ion chromatogram of a biosolid sample. So the dots signs are the polymers that we identified. Like I said, before you do this, you run your polymer standards like we all do for FCR and Raman. So you run them and know the indicator ions that you want to use for quantification. So in our lab, we've selected, and this conforms with literature and what people are also using to actually quantify. So you pick your indicator ion and then screen them in your pyrogram that you get after your analysis. So these are samples, one biosolid samples, and these are the polymers we found in. We found polyethylene, PVC, we found polystyrene, PMMA, and this, like I said, it's just one injection. So we had about 7 polymers. 1, 2, 3, 4. Yeah, 7 polymers. Using the pyrolysis product that we've already identified using standards. Now with polyethylene, what we realized was that because we're not doing a digestion, and because polyethylene has a lot of interferences, when you do a pyrolysis analysis, a lot of interferences are there for polyethylene. Actually, in a way to avoid this, we then put in a strict validation criteria. So the criteria is that usually when you analyze polyethylene, it gives you a triplet peak. So you should have more than 10 of those triplets. Maybe I'm going into the technicalities, but the idea is that polyethylene will give you triplet peaks. The idea is we want to get more than 10 triplets. In real sense, if you don't have polyethylene in your sample, you wouldn't get that much triplet. And then after that, you find the ratios between the triplet, which should be the same as your standard. And then you use a multivariate calibration to actually then get rid of the interferences. So we've been doing this and it's been working well for us. Now there are potential sources of uncertainties. With the main thing is calibration, pyrolysis, limit of detection, oxidation, like whether weathering of plastics can actually impact because we use a virgin polymers to do our calibration curves. But then later we thought about, okay, if the polymers are weathered, would that actually impact quantification? So there's a PhD student actually working on this. I'm not going to talk much about it because it's under review. So they did some oxidation, they did some artificial weathering to see whether this actually impacts whether you use virgin calibrations and you use weathered calibration to quantify using pyrolysis. But I can stress on the selectivity of indicator compounds. Now, the indicator compounds that we use for pyrolysis analysis can actually have interference, like I previously mentioned. In our lab, we had interferences for polyethylene, like I said. What we've done is, like I said, we had those straight criteria in there, and then trying to use multivariate calibration to actually help. Now, what we did was that we picked some organic matrices, like things that we think will produce those indicator ions, and then tested them in our lab to know whether the first shot method can actually get them out. And this is a PON, which contains proteins and stuff. And then the first shot was actually able to get rid of all the interferences. The peak you see here was an internal standard that we spiked to know whether the instrument was working well or whether the instrument was behaving all right. This was fish that we checked. And then the first shot was able to take all the interferences. This was also engine oil. So what we realized was that the double shot works well. The first shot is able to take away all the interferences. So like I said, if you have this, then there's no need to actually do a digestion of your sample. But the good thing is that I only analyze 1 gram of my biosolids and I get about 7 polymers in them. Whilst people doing FTIR and Raman can analyze about, I don't know, I don't do it, so I don't know, maybe I don't know how much samples they analyze. I'll keep it there. And then, so I'll show examples of where our lab is actually at using pyrolysis. So in this work, I'll talk about biosolid because it's what I'm doing for my PhD. But I know we are doing tire particles. We've published a paper in ESNT letters trying to compare the ISO method on tire method because they have ISO has a standard protocol for tire quantification. We had some few issues that we've already published. There's Stacy who is working on road dust. We have a paper which is under, I think it's minor revision from hazardous material, which is on Australian road dust, quantifying the amount of plastic released to Australian environment through road dust. We have a paper accepted from hazardous material, where we quantify plastics in rice. We look at those who wash their rice, those who don't wash their rice in storage container, is it storage container? When you buy rice from the shop and you microwave, something like that. So we did a lab simulation where we wash the rice based on how people treat rice in homes. And then it should be out soon. It's accepted, so it should be out. And then we have seafood, where we quantify plastics in seafood. And then Steve and Tanya are actually looking at surface changes. So they're looking at weathering, how surface of plastics are actually changing with pyrolysis. So I'll just go into what I do. So my first, after getting the method to work and publishing, we then applied the method to biosolid samples across Australia. And what we did was that we took samples from 82 wastewater treatment plants and then quantified the amount of plastics in milligram per gram of dry weights of biosolid. And then the concentrations of the total, we quantified 6 plastics actually. That was polyethylene, PVC, PET, PP, PS, and PMMA. And then we quantified and realized that the range of these plastics, total plastics together, was 0.4 to 24 milligram per gram. Now the predominant plastic was PE, which was like 69% of the total plastics put together, followed by PVC and PET. Now the per capita release of these plastics were actually, of the treatment plants, were 8 to 850 gram per person per year across all the investigated plants. But the funny thing is that we couldn't find any significance between the per capita loads released and the size of wastewater treatment plants, and also between Australian states. And there was no relationship between industrial area and per capita loads. One of the things that we thought was that it's better maybe we get 1 treatment plant or 2 or 3 treatment plants to actually do a mass balance, to actually know where or which stage actually get rid of the plastics. Because what we did in this study was to just take the final output, what was actually being applied to land, to analyze. So that's the next step that we want to do. But this study actually was published in ESNT and is the first study to actually provide per capita mass basis based on local data. And we could only do this because we have a pyrolysis and combined with ACE. Now, this is a paper, it's under review, it's not published, so I'll just talk about it. So we looked at plastics in archive samples from 1950 to 2016. And the one we realized was that it's UK and Australia samples. And then we noticed that the increasing rate of concentrations were actually matching with plastic production globally. So it's actually matching. So production of plastics and the concentration that we found in the biosolids were kind of going up in the same way. Specifically, PE started increasing, I think. So PE was the most prominent one. And then we realized that the production of polyethylene started. So before, from 1950 to 2000 and no, to 1990, we couldn't identify any polymer except polystyrene. Then from 1990, we realized that plastics were actually being identified. And then they were dominated by 3 types of plastic, which were polyethylene, PET, and then PVC. They were actually increasing from the 1990s. Apart from that, we didn't find any of the plastics or identify them in any of the samples below 1990. Now, what someone might ask, okay, so if you want to do a size fractionation like people do with the FTIR and stuff, so you just filter the particles or filter your samples into different sizes of pore size or whatever. How can that also work? So what we've done is that we've tried to couple size fractionation to the pyrolysis GCMS. So the idea is that you sieve or you filter your samples into different sizes. We have a paper that's published in Journal of Hazardous Materials Letters, where we actually quantify the below 1 micron fraction of plastic that's actually been mixed in most studies. So that was on, I think was deployed oysters. So we deployed oysters to the Brisbane River, and then we tried to look at the amount of plastics that has been lost or that's not being reported in current studies to know the fraction that is actually not being reported. And it was the highest concentration was actually from the lower size range. So we applied the same thing to my work, which is the biosolid. And what we found was that, so what I did was that I sieved my biosolid samples into sizes. And currently in literature, the highest particle that people have quantified in biosolid is above 20 micron. So what we did was that we could only sieve particles within using the 25 micron sieves. So I just sieved them. And then, like I said, the concentration of plastics in the below 25 micron range was higher than all the other fractions. And this is only possible because we were able to get the concentration of this size. Now, I've also done it relative to the sizes of biosolid that I got for each sieve, which also matches with the same. So what this tells us is that currently most studies are actually missing a lot of, or underestimating a lot of plastics in this range. Because I think with the data, it's about 27% or 24%, if I remember very well, that's actually being reported under the 25 micron. And most of the current studies on biosolid don't report any of this. They are all reporting particles above 20. Now, as a take-home message or something to summarize with, I would just say pyrolysis GCMS provides accurate mass concentration of selected plastics and can be coupled with size fractionation. Now, the double shot method we have, coupled with multivariate calibration, reduces potential interferences with metrics, potential issues with metrics interferences. Now, initial studies show weathering as an important consideration for polyolefins, like which is polypropylene, polyethylene. But the study is under review and it's not my main work, so I can't talk much about those works. And then compatible with sizing techniques. So currently, my supervisor managed to get us a size, is it A4 size fractionator? It's something that you can use to actually size fractionate samples. Automated, it's an automated one that we just got, we're still calibrating. And then it extends our ability to detect plastics in the nano size range and also all particle sizes. So with the current method, it doesn't matter whether you have smaller size particles in your sample or you have big size. Once you process them and you extract them, you're able to quantify the total plastics in your sample, which is good for all of us. And then this is the team. Some pictures are mixing here. If you want to send me an email or if you want to talk to Holly, anybody you talk to in the microplastic team or PAN-micro team can send me the email of what you want. And I'll thank fellow guys in the lab who helped one way or the other. Thank you.
Thank you so much, Elvis. I will, for everyone who's listening, if you want to raise your hand or just unmute yourself or just put a comment in the chat, we can relay the question. So we've got a good 30 minutes or just less than 30 minutes to chat about the technique. I have one question just while everyone's gathering their thoughts. You've got a very, very nice piece of equipment there. I guess this is two questions. Did the group start working on microplastics before they got this piece of equipment, or was it built knowing that this piece of equipment could be used for microplastics? And then my second question is actually about these polycarbonates. How often do you detect polycarbonates in the environment? Because I know that the industry is quite concerned about the polycarbonates because of the fact that they're the ones that actually have the BPA. So they're quite like, you know, where are they being found? And you don't often find them as well, you don't pick them up as much, but I think you pick them up in some of the biosolids. So those are my two questions.
Yeah, so before we started with the pyrolysis, we used to use the normal techniques. So like I said initially, this is a complementary. It's not like to write off Raman or anything. So we have Raman, we have FTR here. The idea is that we wanted something that can give us mass-based concentration. So currently we have 2 pyrolysis actually. So we invested, I think my supervisor invested so much into it because he wanted to get something mass-based. We have the FTR and everything. So before we bought the pyrolysis, people were using it. We are not the first people using pyrolysis. It's something that people are using and people are trying to develop method. So it's like we started before we bought it, but we knew what potentials that could give us. And we knew what we wanted to achieve with it. So we just invested more time into it. With the second question, what was it again?
It was all about polycarbonates, yeah.
So polycarbonate and polyethylene terephthalate and polyamide. Using pyrolysis was difficult. The tanks, I think Sigma has an alkaline agent, so which you spike onto your samples and increases the sensitivity of these 3 polymers. So currently most labs are using it. I've seen a couple of publications where they've spiked that on top. So we are also using it now. And with the current paper that I have, we're able to spike on top and it actually increases the sensitivity. So with the size fractionation of the biosolid samples, I have spiked that agent on to increase the sensitivity of the polycarbonate and the polyethylene terephthalate. So it works. It's just that you have to spike or put on the alkaline agent to get it to work.
Okay. We do have a comment or a question in the chat. Stephen D. Allen, I don't know if you want to unmute yourself, but I'm happy to ask the question as well. Basically, great presentation, Elvis. This is a great toy. Has the double shot method been published yet?
Yeah, that was the first paper I published. It's already out. So if you can send, if I get your email, I can send you all my papers. Or if you look out for me online or anywhere, they are there. That's what we're using now.
What we can do, Elvis, is when I send the recording,
okay.
We can send the paper with the candidate.
Okay, that's fine.
Any other questions?
I have a quick question that I might have missed. Hi, great talk, thank you. When you went less than 25 microns, what was the minimum again? Was it 1 micron? So
we have 2 studies now. One is published on deployed oysters, and that one we use a filtration setup, and that one we're able to quantify the sizes below 1 micron. So we used a nanofiltration to actually get that fraction, and that was the fraction that constituted or contained much of the plastics, of the total plastics together. With the biosolid, I couldn't use that technique. I could only sieve. So the fraction I could sieve, the sieve size I could use, the lowest was 25 micron. So the ones below the 25 micron, I lumped them together and quantify the plastics in them. And that concentration was higher than the rest, like if you put all the plastic concentration together. But that's not published. It's something I'm still working on. So.
We have another question here from Andrew Mays. I don't know, Andrew, if you want to unmute yourself, but I'm also happy to ask on your behalf.
I can unmute, Holly. It's fine. Hello, everybody. I was very struck in your data that the levels of polypropylene you detected are really quite low relative to many other polymers. And yet, if you look at production levels, they're actually quite high. and I just wondered if you had any comment on that or any thoughts about it. Is it something about the detection levels of the technique due to the actual marker compounds you're following? Or is it just that maybe, I don't know, in Australia, the wastewater process is different, or what people flush away is different? Because certainly we find quite a lot of polypropylene in our samples.
Yeah, so this is something that has come up. And so there were studies before we actually started with the pyrolysis, and they actually found polyethylene terephthalate to be more than even PE. So what we want to do now is that we want to do source tracking. We want to actually get to the treatment. Like I said, we want to take 1 or 2 treatment plants and just do a direct source tracking to try and see where are the plastics coming from. Why is it that we are seeing more polyethylene than polypropylene? Or why is it that, because like you said, you rightly said, globally it's polyethylene that is the highest produced, followed by polypropylene. So it makes sense for you to get that, you know, in that order. But if you look at the plastic, yeah, I don't know if I have that data here. So what we did was that our data actually matches very well with the plastic consumed in Australia. And in Australia, the major plastic consumed was polyethylene followed by PVC. So our data, it matches with the amount of plastics or the plastic volumes that are consumed. Not like to the global one, but to the amount consumed in Australia. That matches very well. I don't have that figure if I get.
But I guess you also need to think about how it's consumed, because if it's arriving in a water recycling center or wastewater treatment plant or whatever you want to call it, obviously it's coming from particular sources and waste streams, which aren't necessarily correlated with the overall
consumption. Yeah, I understand it.
Yeah,
so, so that's what I'm saying.
Yeah.
So it's something I'm writing. We're writing a proposal, not a proposal, but like something to do. We want to look at source tracing, because I think that's the key thing. That's the only gap we have in my paper now, like in my whole story of my PhD. So the idea was to do a source tracing. Like we want to go back and trace what's actually coming in and which ones are coming in, where are they coming from? Why are we having high polyethylene and low polypropylene? So it's something we have on the table, actually.
We actually, Jen, there was a paper done, it's still being written, but we found mainly PVC just after the wastewater treatment plant, which we thought was very unusual. But then looking at some papers, they also found that because of these PVC pipes that are associated with the effluent pumps. So I guess it's really what is in that area. Associated with the biosolids and things like that.
Yeah.
Was that in the water column, Holly, or was that in sediment?
It was in sediment.
Because I'm just thinking about the density here. Because PVC is dense enough to settle, whereas the polythene and polyprop would theoretically at least float and get washed away.
Yeah, exactly. That makes sense. Yeah, I think like you rightly pointed out, I think one of the things that we want to do is that. So like, I understand where Andrew is coming with the question. We just want to find out, like, is it from an industry? Is there maybe a polymer production company sitting somewhere? Like, we just have to find out before we can actually make a conclusion. So in the paper, we didn't try to give reasons for that, just to be on the safe side.
We have a question here from Henk. Henk, I don't know if you mind if I go, because I know you are having internet problems. So I don't know if you want to ask, or I'm happy to ask on your behalf.
Go ahead, Ollie. It's a bit hectic here.
No problem. So he asked, do you find interference with additives such as plasticizers and others? Some of those are halogenated.
Yes. So with a double shot method, initially, when I was developing the method, that was the plan, to try and use the double shot method, to use the first shot to get the additives. But then the focus changed because we were thinking of using it to clean up our samples. So in the first shot, which will then contain the additives and stuff, for now, we've not really paid attention to it, which was my last objective for my PhD. I hope I can finish, but I don't know if I can do that now. But the idea is that we want to go in and look at additives. So currently with the polymers in the second shot, we don't have interference from the additives. But the first shot that we use the low temperature for, that is what is assumed to contain the additives, but we've not really dealt into that. So yeah.
And we also again have a question from Stephen D. Allen, is what is the smallest amount of each type you can detect? So I guess it's the
limit of detection.
Limit, yeah, limit of detection.
Yeah, so there are so many factors. It's instrument dependent. So if our instrument is very clean, you can detect maybe 0. And to be honest, it's polymer dependent. Like polyethylene, you need to put in more to actually detect it. Some plastics like polystyrene, if you put in 0.01, you still see polystyrene. So it's polymer dependent. It depends on how clean the instrument is. To be honest, pyrolysis is a dirty technique. Within a month or two, we clean the instrument like twice. So it's very involving. So all these factors come into determine the limit of detection. But usually we have 0.05 to 50 microgram. That's the calibration range we usually have. So then the detection will be 0.05, which is then dependent on the polymer.
Great, thank you. So do we have any other questions? We still got a few minutes. Just having a look through. Looks like we have another question in the chat. Excuse me if I say your name wrong, but Nska Benson. So if you would like to unmute yourself, or I can ask on your behalf, and you can correct me on how I said your name.
Please go ahead.
Thank you. So you said great presentation, Elvis. What do you consider as the major methodological challenges of using the pyrolysis GCMS compared to other methods of plastic characterization?
Yeah, analytical challenges. Which are the analytical challenges? So personally, I'm going to be more personal here. Most of my challenges has been the maintenance of the instrument and the cost involved. Apart from that, with analyzing samples, yeah, I mean, it's still evolving, right? And it's still an emerging technique. The method that we have might have errors, like especially like I said, with the polyethylene and polypropylene dissolving. Room temperature, it's kind of crashes quickly. So we try to work on all these things. So with analytical challenges, I think it's been fine so far. That's what I can say. I don't think we have a major challenge, because before I started, there were papers I read where they were talking about limit of detection. They were talking about the amount of sample you can put into the cup. But then they are talking about when you want to put solid sample into the cup, because the pyrolysis cup can take, it can take only 80 microliters of a solvent. So just imagine if you want to put a raw biosolid into the pyrolysis cup. In that way, you can't. But then if you have a way to dissolve it in solution, then that becomes possible.
So on that question, because I know we also have a question from Mace, but on that question of this maintenance and expenses and things like that, I'm not aware. I mean, we don't have a database of what techniques are available in Africa, but do you think that there are other people working on pyrolysis GCMS in Africa, and the maintenance and costs behind it and things like that?
So with the maintenance, I think one thing is that you learn on it like as you go, because that's what we do. But I think there's one group in South Africa, if I'm not, because I read their work in is it CTAC?
I think they might have. You're right. I think they might have presented at CTAC Canada
yes,
Canada.
Canada, yeah, yes. So I think they have pyrolysis, and they are doing something similar. With the maintenance and cost, any lab that buys pyrolysis already understand that it's expensive. Like, because you have to be changing column, you have to be maintaining the instrument, cleaning, changing the tubes. So, I mean, you need to have money to actually operate it. That's what I can say. I mean.
No, that's that's the perfect answer. That's what I was trying to figure out.
Yeah.
And Andrew Mays has another question. Andrew, you can unmute yourself and.
Yeah, I was just wondering,
Okay, so what happened is that so like I said, there's a single shot. Most of the labs use single shot, because what happens is if you're using a single shot, then any sample that you analyze with, like all the sample processes you use for FTIR and Raman can be applied. Because in our lab, what we do is that when you have your filtrate on a filter paper, we just cut them into smaller pieces, round them, because before you have them on the filter, you've done your density separation, you've done all the digestion, so you have all your particles. Instead of going to use FTIR and Raman to identify them, you can then cut the filters into pieces, roll them and put them in the pyrolysis cup and analyze them. So in that way, you don't need to do double shot. That's a straightforward one. Just imagine if you want to pick particles and analyze with FTIR, the same particle, instead of picking like 10 of the filter, like people do now, you can just cut the filter into pieces and just roll them and put them in the pyrolysis cup and analyze them with a single shot. You don't need to do double shot or anything.
Right. But in that case, you would have digested them to remove a lot of that biological material beforehand.
Yes.
And then you can get reasonable limits of detection.
So I remember when we were doing Thai particles, I think we analyzed some snow samples for Thai. To be honest, we didn't do digestion. We just did a direct injection. So you just take the snow sample, you put them in the cup, and you analyze directly with a single shot. You don't even do double shot. Because once you are doing Thai way, it's very difficult to get interferences from other matrices. So the interference, like if you're doing polypropylene, it's hard to get interference for polypropylene. So those polymers, even if you have them in a sample, you don't do a double shot, because you don't have interference for their markers. It's easy to do a single shot and analyze them.
Mm-hmm. But you'd never be able to do that for polyethylene, presumably.
Yeah, because there are a lot of alkanes and alkenes. A lot. I think the main issue is polyethylene, and even globally, people are still struggling with the polyethylene interferences, like with pyrolysis, because people are trying to use, either they want to use the alkenes or alkane. Like people are trying to use different markers to just. I remember I read a paper and they were saying that if you use the alkanes above, the carbon chains above C16, C18, C19, C20, then you reduce the interferences from fatty acid and all that. But then if you use C10, C11, they think you have a lot of interferences. So people are trying to get away around the polyethylene interferences.
Mm-hmm. So you think that's a problem that with more work and more careful data extraction methods, you can probably find ways around that? Yes,
there's probably a way, because if you use the bigger carbon, like I just said, it works. It's published already. It's a known knowledge. Like people do that and they go around it. We use the multivariate and use strict criteria. Recently, we were trying to use, so we bought the oils, triglycerol, and we're trying to find a way to actually break them down using an enzyme agent. Like we are also trying, like currently in our lab, we're also trying to see the best way to actually get rid of it as well. Because we had a lot of interference from fish. I think we were trying to analyze fish, and the fish had already gone to KOH, digestion and everything, but there was still interference from polyethylene. Even though they've done the digestion and everything, we thought taking off using digestion to get rid of them was going to be easy. But then we had a lot of interferences. So that's something we're working on now, like trying to find a way around it.
Great, thank you. I think there was a question, I might have muted you, Dr. Neelavan Rana. I can unmute you now and you can ask a question. While we wait, if I misunderstood, sorry, but we do have a question from Andrew Tretwin, who wants to ask about, did you pick up any differences between plasticized and non-plasticized PVC? So can you tell the difference between the different PVC types, if that was something you looked at?
Yeah. So, yeah, I like the question. You know, I remember when I went to US, the same question was asked about polyethylene. So we have low density and high density. To be honest, with polyethylene, it's very difficult to use the pyrolysis to differentiate between the low density and high density. So that's why most of the pyrolysis works, if you've seen published, they try to lump everything together. They call it PVC. They don't try to differentiate them. They call it PET. They don't try to differentiate between low and high because it's very difficult with the analytical technique. It just gives you peaks and you try to make meaning out of it. But then you can't use something to differentiate between polyethylene, low density, polyethylene, high density, soft PVC. It doesn't give you that, like, unless, like I said, it's still an emerging technique. Maybe with time, people will try and find ways to actually get markers that can be specific to soft PVC, something that can be specific to hard PVC. But currently, it's very difficult. And it's not only, like, it's everyone. Like, it's something we know. It keeps coming up at conferences. So
I think he's also asking, because we've recently submitted a paper that had, it's got TGA, FTIR, GCMS, and we don't have definitely the sensitivity that you have with your pyrolysis, GCMS and things like that. But one of the things that it was able to do was tell the difference between the PVCs, which was really useful. But
that's great.
Polyethylene, it was not. You couldn't tell the difference between the low density and the high density.
No, that would be great. To be honest, that would be great. That would be great. If I'll be happy to read the paper, because if there are specific markers, you know, we have a TGA, we have a TGA. So initially we're trying that method, but we don't have it coupled to GCMS. It's just TGA. So we're trying, but then we're struggling. So we stopped. Like, we couldn't continue. Yeah. So it's good. To be honest, if you have it, that would be great. I would love to read it, because that will actually help me to actually then also separate it.
Fingers crossed we get a response back from our reviewers soon, and we'll be able to share that.
Yeah, that would be great. That would be great. To be honest, I would love to read it.
Well, if there's any other burning questions, we still have a few minutes. Otherwise, we will probably wrap it up. And I think I'm just checking for any of my announcements. Before we close, if we can just remember that if you do have any specific topics that you would like the group to address in the future, please do let us know. You can get hold of Elvis, myself, Athena, or Fedara. Then we can try get someone to speak. We also really welcome anyone from African-led research and African universities to give talks about their experience and things like that. This is showcasing work being done in Africa. So if you feel like you would like to give a presentation, please do just get hold of us. And yeah, thank you so much, Elvis. That was really, really helpful for all of us. We're all very jealous of the equipment that you have there, but it's really good to
when are you buying one?
Yeah.
when is your lab buying one?
We're trying.
Yeah, if you have a GCMS, I think if you have you already have a GCMS, right?
Yeah, shame Andrew Chetwin is currently on the call, but he can't unmute because he's in the lab. So I think he wanted to chat to you a bit more, because he recently read your paper. I think the one that you mentioned on the oysters.
we have. So yeah, I mean, if you have a GC, I mean, if you get like 20, I think 80K or 100K, you should get a pyrolysis.
You should just get an add-on. But yeah, if you can share some of those articles that you'd be happy for us to share with the group, I think that would be really nice, because then we could have access. And yes, thank you again, and thank you everyone for joining. Yeah. One
thing I was going to say is that I don't know, you know, how I know we have a lot of people from Africa, like who are in Africa trying to do some of this analysis. What people have done is that they try to contact my supervisor or whoever, whether it's Raman or something, to try and see if they can ship samples. Because if you don't have it, like one of one good thing about this platform was to try and see how we can also try and, collaborate. So if you have samples that are really something that will be interest to other collaborators, I mean, I think Holly herself will definitely be interested in someone brings samples. You have your work, you want someone to analyze something to just make your work a bit tougher, because I've reviewed a couple of studies from Africa. And to be honest, one of the things that comes up all the time in reviewers' comment that I also say is that I know technologies are improving. People try to use needle and stuff to quantify the plastics. To be honest, when you submit it and it comes to people over here, it's a total rejection, unless you go to maybe a low journal. Once they see it, they don't care whether you are in Ghana or something. What they care about is that they want you to collaborate with others. So if you have a method, you want to use needle to identify the polymer, you can collaborate with someone, get some few samples to the person, 10-5, some percentage, and the person use Raman or FTR to just confirm for you, just to make your work more, standard, like compared to others done. Because I've seen a couple of them and they try to use needle and they just say, oh, we don't have access to the instrument. But these days, people are not buying into that. They just reject it.
I think that's a really good point for everyone out listening and part of our group. I think that's what we're here to do is really link you with people that can help you get the work that you're doing published and recognized, because there's a lot of polymer identification is one step, and we don't want that step to hold you back from actually publishing the really good data all the work that you've put into the work. So I will share, Elvis, I will share your contact details when I share the link and everything like that. And please do get hold of us if you're interested in finding ways for your data or your samples to be identified with polymer identification, and we can try and link you with other groups around the world. So yeah, that's a really good point. Thank you.
No, thank you.
Great. All right, everyone.